So my talk's going to cover a good bit of ground, and I really, I think a lot of the previous speakers set up a lot of really interesting topics, and some of this is actually kind of things diving in a little bit into the science or the data and how we handle it and how we really want to. I think the CSS is really chemical safety for sustainability. It's a research program implemented within the Office of Research and Development, and the issue there is or what it's trying to do is really integrate. So I come from a small center, 20, 30 people. Focused on computational toxicology, and you have a lot of exposure scientists, health effects researchers, and so the real goal is integration. And the dashboard, I think, is that main mechanism, or at least the grand goal is that main mechanism for pulling all that research. And I think really the way I say is quit stopping at manuscripts. So we hear about unstructured data, about all this. I think the goal here is to not stop at a manuscript and really try to take models and tools and data and knowledge and move it forward. So I'll give a bit of a background, kind of the problem statements, kind of our initial one, and kind of where we're at now. Quick overview of the research programs. I'll try to do this quickly because I think based on looking at the room, I think most people have a decent background or I've heard talks about some of this. One area is that our high throughput screening data analysis, kind of the workflow, the pipeline for analyzing the data. And I don't do it just to kind of give an overview of the data, but some of the complexities and why, both just shoving out raw data or just shoving out hit calls, of yes or no, and what dose or concentration does it happen, doesn't really cut it because I think of a lot of the points people have brought up already. You really need to put this data out there, and prescribing an answer isn't the answer, and also just saying, here, have fun with this data. We're giving kind of background on this information, and so kind of going through this pipeline, I'll hopefully demonstrate where people at different levels might want to come in at this, and then moving this as just one example of research moving into a dashboard, and then kind of how this can be generalized, not just for kind of, well, in a relative sense, pretty homogeneous data to the very heterogeneous data that's out there in the environmental health world. So the problem statement, I think this is key in this discussion of kind of emerging data issue because I think a lot of folks in the environmental health world kind of forget that at least one of the biggest problems is kind of moving away from the descriptive and the lower throughput, the few risk assessments a year, a few chemicals, probably 80–90% of chemicals really having no information on them, and kind of get away from that and want some sense of truth. And I'd argue there really isn't much sense of truth in just say toxicology. 50% of chemicals cause rodent tumors. Does that mean 50% of chemicals cause cancer? Or have a human, have human relevance. And so I think there's a lot of mechanistic research to be done. But really, at the end of the day, all of this just really comes down to probably a probability of a chemical having some kind of negative impact in a disease state. And so these are the problems. And what we've done, at least within the CompTox Center, is say, okay, well, what we can do is produce a lot of data on these chemicals, and that's kind of created a new problem. And being born in the 80s, this is kind of the knowing's half the battle, and kind of your red lasers, blue lasers are all the data. We spent a lot of time generating this data, and now it's come down to where we're trying to create knowledge from this information. And we've spent probably the last 5 years, in at least the ToxCast program or in the high throughput screening data generation, there a good bit of time in the knowledge development, but really a group of 20, 30 people, or even just say all VPA, 500 researchers or whatever, aren't going to be able to turn all that data into useful. So I think part of the dashboard project is also putting that data out there so that other folks can chew on it and kind of improve the knowledge base coming out of this data. So just because I think there's a lot of confusion sometimes with the ToxCast and Tox21 efforts. Both are data generating programs attempting to produce a lot of high throughput screening data on a diverse set of chemicals. The main difference between ToxCast and Tox21 is really in kind of the scope. So ToxCast does hundreds of chemicals, now up to about 2,000, but across hundreds of assays. Whereas Tox21 generally focuses on thousands of chemicals, but across about tens of assays a year. And so that just kind of gives you the little bit of the scope on the data that's being generated. And ToxCast is a little bit, has links to a lot of program office activities, and just being kind of housed within the EPA. It has a little bit of, a direct purpose for some of that, whereas Tox21 is a big interagency project. But they do generally share common goals. And I kind of gave a real quick intro, but I really just want to point to, there's a lot of information out there on the research program, and I think it really does deal with trying to make our research within the agency much more integrative. And I do see the dashboard as kind of, this interface between the research and the public, and then even with our program office partners. So a lot of what we do kind of faces two ways, kind of internally and externally. And really at the end of the day, we really hope this to be a customizable web application. Dashboard is kind of a somewhat of a misnomer in at least IT terms. But really what we're trying to develop is a real customizable web application. This provides some specifics to the data that we're dealing with, and this really just kind of reiterates the thousands of chemicals for Tox21 versus a smaller set of assays and a larger set of assays across hundreds of chemicals. And since we had such a great discussion on ontologies, here's a collaboration with the University of Miami Bioassay Ontology Group. And actually, I agree kind of on both sides. And I think one thing we get hammered with a little bit from a, we'll produce something and the assay title might be, ATG underscore, cis or something. And everyone says, well, what does that mean? And, it's tough to write descriptions that really mean something to, multiple levels of people, biologists, computer scientists. And so really the goal here is just to be descriptive and try to be transparent and try to put out information. This is one way I always say that, with this type of thing, this is one way of describing the 1,000 different readouts, 700 different assays that are getting pumped out of this. It's not the only way. How you describe the intended target of an assay can be very complex. And so we just do our best to do that. And really what our hope is people can come in and have different views on that and be able to kind of, supplement what's already been done. One thing is kind of the redundancy in this is a little, well, maybe not too tough, hopefully not too tough to interpret, but basically we map all these 700 assays to a gene. And one thing you'll see is that there's a pretty good redundancy across many. So this is, greater than 6. There's about 4 different genes with more than 6 different assays in the ToxCast program. And as you move up, there's about 200 genes with only 1 assay covering it. And I just bring that up because I think, there's a lot of focus on some really well-covered areas of biology with, lots of assay technology, but kind of as a call for more data generation or more assay development, there's a lot of areas of biology in which, the high-throughput screening maybe covers, just doesn't cover enough. Okay, so with all of this data, I think this is a new issue. So many of the, kind of the genomic data comes in. I think there's a lot of really well, described methods for analyzing genomic data. And a lot of the high-throughput screening data, I think when I first came on, I was like, oh, go talk to a few pharmaceutical companies or a few, whoever, and they'll have all the answers. They'll be like, oh, just do this, this, and this. And what you find is, kind of, and we have some great collaborations actually with, a whole suite of pharmaceutical companies. And what you find is, a lot of the assay batters that they use, are kind of specific. They can be a little bit siloed. The way the data is analyzed can be pretty different. But what we realize is we have hundreds and hundreds of assays. And so, and the data comes in across, 15 different, platforms or 15 different technologies across many years, and in many, many, many different formats. And so what we really figured out was, well, shoot, we actually have to do a lot of this data analysis pipeline ourselves. And at first, what we did was say, okay, postdoc 1, postdoc 2, postdoc 3, here, analyze this data. Well, at the end, and that was kind of our phase 1, one of the big lessons learned is, well, it just made data integration that much more difficult. And so even though the value of that information, was high in some really nice manuscripts and a lot of good, biological studies coming out of it, but really then trying to put it all together, some of it was a little bit apples and oranges. And so this pipeline, I think, hopes to try to get through that. And what you see is there's lots of, 27 million data points, 1 million chemical assay concentration response curves. So that's a lot of, combination. So we developed a 8-level process that goes all the way from kind of these raw data formats to basically uploading into databases and eventually out through the dashboard. And like many folks have mentioned already, is the real goal is to put this data out across that entire pipeline and to make all the software, the underlying scripts and data analysis pipeline available so people both can extend it, they can modify it, do all sorts of different things with it. This is a summary of the data analysis pipeline, kind of all 8 levels. I won't go too deep into it, but I guess some of the points that I'd like to make with it is it's not like pharmaceutical companies or other people have been analyzing high throughput screening data or other assay data. They had every single piece in this, meaning someone had thought about normalization, someone had thought about background subtraction, all the way down to how you filter cytotox. It was really about trying to, kind of leverage all that information and put it into one system. And we're not the only ones that have done, especially up to what I would call level 6. So all the processing, we even a lot of our partners in the Tox 21, they have very similar systems of getting the data all the way up to where you basically have, some sort of curve fit going through the data and a yes/no of whether you think it's a significant hit. Some of the differences, I would say, is the diversity of data. So we're getting data from 15 different, collaborators, contractors, and so the raw data formats coming in, varied widely. And if you noticed on the assay ontology side, it's everything from RT PCR to ELISAs to fluorescence assays, luciferase assays, all different types of technologies. The area I think is really important though is in this 7 and 8, but definitely the 7. I think a lot of it's a PubChem. I think a lot of that data gets pumped. It basically stops at level 6, so it's the data, kind of the line through the data with a hit call. But really, I think what is important, and PubChem has this functionality, but is bringing in other data and do systematic alterations to make that data more meaningful. So if you look at any assay where you run 10,000 chemicals through, you're going to have fluorescent compounds, you're going to have cytotoxic, cytostatic, protein synthesis inhibitors, all those sorts of problems. And I think layering on that information and making that transparent helps kind of the whole community, because what will happen is if I hand someone a level 6 equivalent, basically a yes/no answer just based on that data, there can be a lot of misinterpretation. There can be a lot of folks coming in using that and pumping it right into a model, and I think it can get a little bit misused. And so I think it doesn't mean that level, kind of a hit call level is not important. It just means that you have to have it in the context of kind of the entire pipeline. And so I think it's one big change that internally we're going to do, but I'd advise kind of more generally is that kind of, it's not just releasing raw data, and like I said earlier, not just releasing your end state data. I think it's kind of everything in between with some context around it. And here's just some kind of real world. Be great if everything was flat or looked like this. Pull some summary statistics from it, but you have things like baseline issues, noisy data, single, what happens when all you see is a little bit of activity at the end. You have outlier detections. A lot of this stuff has been prescribed. Cytotoxicity. How do you handle that? Do you really believe that? So there's a lot of interpretation within this high throughput screening data. But the important point is there's no single right answer. There's no one assay that is perfect. There's no assay that produces the right answer every time. They all have confounders. They all have issues. And so you really need to kind of take a little bit deeper. And I think probably the biggest question is this statistical versus biological? And I think the genomics world struggle with that, with, you know, whether twofold induction is meaningful for one gene versus another. And I think that same kind of thing happens here where, you know, a 20% increase in some activity in a cell-based assay, you know, what does that mean biologically? So just to give an example of something that came out, and I don't know if it would have come out in the kind of first way that we went about this and this kind of lessons learned session. So I think the first time it would have been tough to pick out on some of the patterns that you see in the data because of the analysis approach was a little bit more siloed. But I think now that it's in this pipeline, I think here is just an example of protein fragment complementation assays where we screened about 2,000 chemicals through. And what's interesting here is these green ones are all reference antagonist assays. But this assay was set up solely as a, or was, you know, kind of an agonist format, meaning there was no estradiol induction looking for a suppression or anything. What would happen is you actually kind of get this super maximal activity of all of these, and, you know, nice separation. And here you have a lot of, in this, right around this 100% range, you have things like your steroid hormone, so estradiol. And so you have this nice spectrum going from here, something like a bisphenol A would be kind of in this middle group, kind of a partial, well, the way I'm interpreting it at least right now is kind of a partial agonist moving to antagonist, and something like raloxifene, you know, a more pure antagonist is over here. And the point isn't to bring up all the biology. I think the point though is if all you did was you know, release Emax values, which would be, you know, these values, the maximal response relative to estradiol, that could be misinterpreted because that might be called, you know, an agonist ER alpha assay. And so I think there's a next layer that has to kind of characterize these, or chemicals in the library that act more like this than these estradiol steroid hormones. You have to put that into some context. And I think that's what this pipeline will enable, and hopefully through the dashboard that can be, you know, illustrated. So, you know, we have some relational databases, but I think where we're going is in this web service technology, which is really going to allow us not to be wed to any database and can hopefully integrate data across databases. And so it won't be just pulling data from ToxCast, and a web service is really kind of, you can think of it as a URL that goes and goes to, you know, can go to almost any database, and based on, you know, that kind of, if you've ever seen a CAS number, you know, at the end of a URL, that's, you know, a simple way of thinking of a web service, and it pulls all the data for that CAS number. That's an example of what we'll have set up for ToxCast DB and a bunch of other kind of federated databases within and external to the EPA. And hopefully what we'll be able to do, at least initially, is some real simple things like view, you know, all the assay data for a chemical or a group of chemicals, view chemical data for an assay, so view all the data across an assay. So very simple, but, you know, I'll show you kind of down the road kind of how this can get more complicated and how the functionality hopefully will really grow. And then a lot of selection. There's a lot of metadata for any chemical. There's structure, use, so on. There's a lot of metadata for an assay, and or piece of data. So piece of exposure data, occurrence data, hazard data, a lot of metadata. So I think the selection criteria is also really big. And then the end goal for much of what we do within the agency in terms of trying to, you know, at least initially, is using this for prioritization. So integrating hazard, exposure data to, and other types of data to, you know, basically rank order chemicals for further testing, basically making chemical testing decisions. So the CSS Dashboard concept it came in, it was actually kind of initially thought of as a way that we can kind of put 2.0 versions of all our existing databases. So, you know, we get a lot of requests, oh, I can't search by this and this or that. And so we're like, okay, well, we kind of need to think of a next generation way people can access the data. But then it kind of came into, you know, service, a much higher level of service. And I like what Titus mentioned, which was kind of, this was going to be the main, you know, everything should flow through this and everything out through this. And everyone should get data through this. I think that's the grand goal of this. This shouldn't be where someone comes to me and asks me to write a SQL query and I send them a text file type of thing. I think the grand goal is that this will work all through this, and one, I think that'll be much more efficient, but also I think it kind of adds to that overall transparency and kind of data sharing goals. And so the design concepts here is to be highly transparent, very retraceable, so allow users to basically kind of start and stop and go back, updatable, interactive, modular, all the buzzwords. But what we're really trying to do is leverage all the resources internally and kind of going back many years of all the tools that have been developed. So models, EpiSuite is a good example of a agency developed set of phys-chem parameters, modeled phys-chem parameters. I think it's a good example of a tool that really would be needs some integration. So it's a great resource, but it'd be nice if you're pulling that alongside a lot of other data. And right now we just have kind of 5 different, well, I broke it down into three different design concepts. So you really have data selection and exploration, chemical selection, exploration, and kind of this prioritization. And when I say prioritization, this is very loose terminology. I mean, this could be all the way down to combining things and getting reference dose like values. So it could go all the way to that end to very simple adding up hit counts and sorting your chemicals. So prioritization is really just looking across both chemical and data. And so here's the conceptual framework for this, and I think in the theme of kind of lessons learned, what we first thought was, this link here is kind of like your web services. So this is able to pull from many different databases and pull that data into not even necessarily a common format, but I just think of the world, at least in the concept of this dashboard, as chemicals associated with data. So really you can take all these highly structured databases and really it's a chemical, it's a data, and it's a value. And then across all of those chemical and data, you have metadata. And so that's where you kind of break down the relation, kind of these highly relational models and things like that. But I think it generally works because really what you end up wanting to do is going in here, and here's just an example where, just say as a user, I want to basically take this whole entire kind of world of data and bring it down. I only want EPA and NTP data. I want only the good data. Hazard, I want in vivo and in vitro and hazard and exposure type of data. And so with all that metadata, you basically get your kind of entire set of rows. And likewise with the chemistry, you can break it down by availability, by structure, by property. And so adding all this functionality isn't necessarily trivial, but I think the concept generally holds for basically being able to divide up your space. I think one of the big kind of criticisms we get from a database like Actor, which is kind of a grand aggregator of data, is I search a chemical and I get it's a HTML page that if you print it off as a PDF would be 40 pages or something. Well, I don't know what to do with that necessarily, and I think the real goal here is to put some of that in the user's hands so that they can start to decide what data they want to look at. And this goes back to the curation piece. I think the rate limiting step here is curation, not just, okay, we have this data in a database now. It's curating it to quality, to other metrics. I think that's a real big piece that we're constantly, as a group, working on. I think generally people need to work on because I think data quality and other kind of metrics are really important for allowing kind of user-driven functionality. So from there, you basically have defined your data classes and your chemical space, and this is where kind of the fun stuff comes in this kind of dashboardy concept of developing widgets. And it was great to hear the bringing up of Git, and I think this is where Git would come into play down the road, both internally to the EPA but also externally, is in widget development. So there's data. You have sets of public web services, meaning you can go out and you can basically pull data, and you have a nice idea for how to present that data, how to make that data interactive. Well, as mentioned earlier, Git would really allow, in the right environment, for you to branch off, begin to take that data, develop it, and then basically re-enter into this kind of pipeline of the dashboard. And so I know as a small group that's trying to develop this, it's impossible for us, to develop, 500 different widgets for all different data types and to service, every single program office in the EPA and the public, and all different levels of the modelers and toxicologists and so on. So I think that kind of mentality of developing this framework and hoping this framework holds, and then allowing kind of the user community to dive in. And so it is a working, real thing. It's a very alpha version. And so here's just, kind of the same concept. So you have your chemicals here, so the columns from before. Here are your data classes, so those were the rows, or at least the different colors of the rows. And then with any one of these, you'd have the data, so that'd be the individual rows here. And then many different ways. So you can look at things as simply as a table, or complex like spider plots and concentration response curves or whatever. And so this is kind of the main structure. And an example of how we've been able to quickly basically plug in an existing tool, and this is kind of off that Git mentality of the ToxPy. So David Reif, who recently moved to NCSU actually, he developed ToxPy, and it's really just a nice way of visualizing and aggregating data across many data domains. And here's just an example, and this took maybe a week's worth of development basically to take the existing application and basically plug it into this. And really what you're doing is you have your data classes, so here's estrogen, androgen, thyroid, and the user can drive the weighting. The user can play with this. The user can benchmark this against what they think they know or do know about chemicals in their library. This is kind of repeating kind of what I've gone over. I think some of the main points here are that we're hoping to by September 2013 kind of have a more fully functional version. But by SOT, since a lot of folks might be at SOT, we're hoping to have a real early alpha version, at least at SOT, for people to see and kind of get feedback. I think that's where we're at now, is more at a feedback stage. But I think it'll be a good opportunity for us and maybe any potential users. And then a lot of what we're doing, and I think what we're hoping, is to provide program office specific dashboards. And I think the point to make here is that we're not building 25 different dashboards. It's really one dashboard which allows this kind of customization really on just the data that you're viewing. And so that's why I think we can actually get it done. And it's not a kind of pie in the sky type of thing where we think we're going to build a bunch of different software for everybody in the world. So the basic conclusions here are that we really leveraged what was known out there in the pharmaceutical world and other for analyzing high throughput screening data, but really with the scope that ToxCast, or the breadth of assay space in biology, we really needed to come up with kind of our own data analysis pipeline, and hopefully as a result of that, we'll be able to not just be quote unquote transparent in giving out the data, but it'll have much more context around that information. And then that the public release of that data, just kind of as a general interest, is hopefully that'll really coincide with the release of a beta version of the dashboard. And so this will be a main mechanism. So it won't be hopefully just a bunch of Excel files being pumped, or CSV files pumped out to everybody. Hopefully it'll be kind of as part of this web application. And so for everybody of all backgrounds will hopefully find it useful. But definitely open to feedback. And just thanks to everyone in our team, and very good group of collaborators. So.